So Ellen, thank you very much for coming. I'm sure that we're going to have a great talk. And now, talking about my dear friend Allain. Allain, or Dr. Bueno, works at University of Worcester. He started to work in Worcester after a 4-year period of postdoctoral experience at the Institute of Brain Chemistry and Human Nutrition in London, working with Professor Michael Crawford, one of the pioneers in fat acid metabolism and brain composition. And in Worcester, he works at the Biological Science Department, and he is also a scientific advisor of the Food Standards Agency and course leader of this university, the University of Worcester. So thank you very much for attending our conference. And now I'll let you.
It's on us.
With you.
There we go.
With you. Sorry.
Thank you, Monica. Thank you, everyone. Ellen and I are very grateful for this invitation. Thanks for inviting us to talk with your audience, your students today. It's a great pleasure, a great honor. Yeah? Yeah. So today, Ellen and I will be talking about lipids and the way lipids are so important for cell signaling, cell survival, proliferation, differentiation, and also as fuel, energy reservoir. And there's a variety of, like, a huge family of lipids, and each and every one of them with their own very specific function and their importances. Yeah, and Ellen and I will deliver this talk today. I'll be passing to Ellen, and she'll be returning back to me. We kind of push it against to the other side all the time. So we'll be playing some sort of like tennis here, passing the ball one to the other. Okay. Right. Okay. So I will start with myself. Just turn a little bit. Okay. So I will be just turning this screen like this, and then when it's Ellen's turn, I'll move to her. Where's the PowerPoint?
The share screen.
Okie dokie. So there we go, folks. So once again, good afternoon. My name is Allain. My colleague Ellen here is sitting just next to me, and today we talk about lipid classes, exploring the intricacies of fatty acid storage and usage in the body. That's myself here, and this is my colleague Ellen. Yeah, so we are very grateful to the Unifest, São Paulo Federal University. We are also grateful to FAPEST, has funded a significant amount of research, Monica, with several projects with FAPEST and also CAPIs. And we're very grateful to the University of Worcester as well for giving us a lot of support. Right, okie dokie. So talking a little bit about myself, I graduated from Paulista School of Medicine in Brazil in quite a few years. It was like in year 2000 when I graduated. I graduated as a biomedical scientist, medical modality. And then I did my master's in São Paulo. And then for part of my PhD, I went on to the University of Liverpool, working with Professor Paul Triglycerides. And then I just returned to Brazil to defend my thesis. My supervisor in Brazil was Dr. Claudia Oller. You guys may know her very well. But there we go. And then I returned to England. And then I had a 4-year position in London at London Metropolitan, working at the IBCHN, Institute of Brain Chemistry and Human Nutrition. And then for the first 3 years, the IBCHN was based in London at London Metropolitan University, but then it moved on to Imperial College London. But then obviously after 4 years in London, I said, well, enough of London. So I applied for a position as a permanent lecturer here at the University of Worcester. And I am also a registered nutritionist with the Association for Nutrition. Yeah? So for those who don't know Worcester, right? Okay, so I know that many of you here in the audience maybe don't know Worcester. Can I just ask a question in the chat? Have you guys ever heard of this word, this city in England called Worcester? If you have, please say yes in the chat. There is one answer here. Monica's, but there we go. There we go. Right. Okay. So let's carry on then. So we are here now. Remove this. Sorry guys, I'm battling with IT here. Right. Okay. There's London over here. So this is the English channel. Yeah. France would be here on the side. And then London, as soon as we drive up, eventually there is Worcester here. Yeah. So this is exactly where we are at the moment on the map. There's Birmingham here, the second largest city in England. London, Birmingham, Worcester. Yeah. So there we go, folks. So this is my webpage on the University of Worcester website. If you would like more details about me and about my publications, you're welcome to visit my website and learn a bit about my history. I also work for the Food Standards Agency, which in Brazil is the equivalent to Anvisa. Yes, for those who know Anvisa in Brazil will know what they do. And I work at the Additives, Enzymes, and Other Regulated Products Division. Yeah, so there's the work that I do for the FSA. It's a very nice job. It's very important, very interesting, and I've learned a lot in the past few years. And the FSA has become more important over the past few years after the United Kingdom became independent from the European Union. Yeah, for those who know a bit of history and geography, know that until 2 or 3 years ago, the United Kingdom was part of the European Union, but then after referendum, the British people voted to leave the European Union. Until then, we were under regulation of the European Food Safety Authority, but then after Brexit, the Food Standards Agency gained far more autonomy to dictate over what the British people eat in this country. Right, and this is my colleague Allain. That's your turn now, Allain.
Yeah, swing back to me so you can actually see me. And
I don't know how to make that thing.
no. Oh, anyway. Hi everybody. I'm Allain, and I'm a full-time PhD student here at the university, as Monica said. So for me, I'm not originally from the UK. So I actually come from a really small little town in the west of Ireland, and I came to Worcester to do my undergraduate degree in human biology and human nutrition at the University of Worcester in 2012. We're just going to see if we can get rid of.
It's not very nice. If I hit the green thing, so what does it?
There we go.
Okay.
Maybe. Oh well, it might work better. So I was here for 3 years, and I worked under Dr. Bueno for my final year project. But once I graduated, I went back to Ireland, but this time to the south of Ireland. And I did my master's at University College Cork in molecular cell biology with bioinnovation. So bioinnovation is the fancy term for business. So we did business, marketing, and also intellectual property law. After I graduated there, I returned to Worcester first as an associate lecturer in the biosciences, mostly focusing on physiology. And then when the opportunity arose, I took up the PhD scholarship under Dr. Bueno again for nutritional biochemistry, and this is where I am to date. So again, just like Alan said, you can find me on the university's website if you want to read more about me and my work. I come under the Worcester Biomedical Research Group, and my PhD project really focuses on plant polyphenols and fatty acids, and looking at the kind of the molecular disturbances caused by both obesity and neurotoxicity. I work quite closely with Monica's group, with her student, Dr. Bruna Harata, but also with Dr. Eliana Ribeiro, with Dr. Walter Bouldering too, who's a visiting collaborator. So I'm going to start off today just going through the kind of the basics behind lipids. For those of you who don't know much about lipids, I'm just going to go through it with you so you've got a good understanding from for the research that we do. So we loosely define lipids as organic compounds that are insoluble in water and soluble in organic solvents, depending on the polarity of the solvent. And for the most part, we say lipids are insoluble. There are a few that have some solubility, but for the most part, we consider them insoluble, and that's why they float on the top of water when you add drops of oil to it. They have key biological functions within the body, mostly for energy storage, for insulation, but also protection of our organs. They also contribute to the structural components of our cell membranes, and they participate in the signaling pathways within our metabolic processes within the body. So the study of lipids is termed lipidomics, and it falls under the kind of umbrella of the metabolome or the metabolomics, which you've probably heard of. So it's a subsection of metabolomics, and it's really looking at the complete lipid profiles within the cell, the tissue, or the whole organism. And the study of it really has helped kind of create biomarkers and understand the background of some diseases such as obesity, atherosclerosis, stroke, hypertension, and diabetes. So lipids is a big branch umbrella term for a great variety of diverse compounds. They vary greatly in their structure, but their structure gives them their ability to function, and it facilitates the type of function that they have in their body. There's a lot of biosynthesis pathways that occur within lipids, and so they can be built up or broken down, and their intermediates can be used for many, many different metabolic pathways as they go through their biochemical transformations. So because it's such a big umbrella branch of compounds, there's organizations that have now started to really focus on building the kind of the comprehensive classifications, the naming, and the chemical representation of lipids, so that it can be universally named throughout the world. And we're working on a kind of a set kind of naming and representation of them. So this organization, Lipid Maps, is a great resource. It's open access, it's community driven. So if you're interested in it, go check out their website. There's lots of resources. And so just for instance, this picture I've taken from the website, and it really shows you the different types of classes of lipids that they've really worked on naming and identifying. So for instance, we've got fatty acids, eicosanoids, glycerophospholipids, and don't worry about these terms right now. I'm going to come back to them in the later slides. We've got cardiolipins, glycerolipids, as well as cholesterol esters, sterols, sphingolipids, and prenols, just as a kind of a see that there's so many different types of class of lipids that we can look and identify. So for us, there's certain classes of lipids that we are interested in to look at within the samples that we have from our research. And to start with, we tend to look at the total fatty acid profile. So the extracts we get from the cells or the tissues, say from animal studies, we look at the total fatty acids. But that doesn't always show us the dynamic differences that are happening within the different classes of lipids. So all of those different classes that I talked about in the previous slide, we just see the total profile. And if we have a look at this picture just briefly, so Elsterat in 2020, they looked at the adipose tissue of humans, and what they saw is that about 65% is represented by triglycerides, a bunch more is by phospholipids, and then you've got free fatty acids and a number of different things here. So if you look at the total fatty acid of adipose tissue, you might really only see the triglyceride because it's such a main part of it, and you miss the little intricacies of the phospholipid changes. And so this is where our research has really branched out to for our samples. So we like to look at the free fatty acids, the triglycerides, the diglycerides, the monoglycerides, the cholesterol esters, as well as the phospholipids. And I'm going to go through these just briefly, just for those who are really new to lipids. so you can understand what they are and why we might look at them. So free fatty acids are they're chains of carbon atoms attached to hydrogen. They have an acid group at one end and a methyl group at the other. So this is what we name a fatty acid, and they can be completely saturated in that all of the carbons are attached to hydrogens, or the carbons can start to have double bonds where you can start removing some of the hydrogens, and this gives it flexibility. So particularly if they're, say, in the cell membrane, it gives it that extra flexibility so it can move. And so depending on the amount of double bonds that it has and the liberation of the hydrogens, we get more flexibility with it. So we name them saturated fatty acids or unsaturated. And if they have more than one double bond, we start calling them polyunsaturated, so multiple unsaturated parts. So we name them by the amount of carbons that they have. So this structure here is a fatty acid, and it's got 18 carbons. So we call it C18. It's got one double bond here, so we then say 1, and then the double bond starts at the 9th carbon from the methyl end of the fatty acid. So we have C18, 1, N9. So if you ever see fatty acids written down, you now know that it means C, whatever number it is the amount of carbons. This number here is the amount of double bonds it has, and this number is where the first double bond occurs. So we then go into the glycerol lipids. So these are fatty acids that are attached to glycerol. So you have monoglycerides, which have glycerol and one fatty acid, diglycerides, which have 2 fatty acids, and triglycerides, which have 3. So they are formed predominantly either from the food that we ingest in our diet, which is broken down in our digestive system and passes across our intestines, or we can manufacture it predominantly in the liver and then transport it around the body for energy utilization and storage. But the breakdown and build up from glycerol into mono, di, and triglycerides really is a backwards and forwards process. It's occurring, we're liberating fatty acids and then putting them back in, depending on our need within the body. And again, same with triglycerides, we have 1, 2, 3 fatty acids attached to the glycerol, but in terms of the triglycerides, they're really long-term storage for the body. So again, like we saw in that previous picture, adipocytes and adipose tissue can be made up of like 65% triglycerides, really for storage and energy utilization in the organs that it's attached to. But we also, it's used for insulation and protection of the organs. So it acts as cushioning, that adipose tissue acts as cushioning for the organs. And in terms of what we ingest, a lot of saturated triglycerides come from animal products, and if we get unsaturated ones from the plant-based. With cholesterol esters, it's made up of a fatty acid group attached to a cholesterol at the hydroxyl group of the cholesterol, and it's really designed for the transportation of cholesterol around the body. So cholesterol is slightly soluble, mostly insoluble, but slightly soluble, which actually is a problem when we transport it, because it tends to sit on the outside of lipoproteins. But if we attach a fatty acid to it, it creates a very hydrophobic molecule, which allows it to then be inserted into the inside of a lipoprotein, which I'll show you a little later on. But the whole process of cholesterol esters is really about efficient transport of either dietary or synthesized cholesterol to and from the liver to the peripheral tissues and back again. Now, it's made by 2 different enzymes. So in the intestines and liver, we have ACAT2, which is acyl coenzyme A, cholesterol acyl transferase 2. We'll just call it ACAT2. But in the peripheral tissues, we use lecithin cholesterol acyl transferase LCAT. But again, they're doing the same job. They're attaching fatty acids to cholesterol for its transportation. And then it's hydrolyzed back into free fatty acids and cholesterol by cholesterol esterase. So I was telling you about the lipoproteins. So these really are our main vesicle for transporting lipids around the body. It's got this surface monolayer made up of free cholesterol, but also phospholipids, which I'll come on to a little later. And inside we have this core that really is kind of like a vessel for the transportation of triglycerides and cholesterol esters. So going back to what I said about the cholesterol esters, if it was just cholesterol we were trying to transport, it would just sit on the outside here. But now that we've made it into cholesterol esters, it can sit in this big pocket and it can be highly efficiently transported around the body. And lipoproteins vary greatly in structure. We've got HDL, which is high density lipoprotein. I tend to remember it as healthy density lipoprotein because it's the one that actually protects us against atherogenic and like atherosclerosis, whereas all of the other ones, so low density lipoproteins, intermediate and very low density lipoproteins, these are all pro-atherogenic. And these really are transporting cholesterol and the fatty acids and triglycerides out from the liver out to the peripheral tissue, whereas HDL is bringing it back in for utilization. So, and finally, we have phospholipids. They make up the main membrane of our all cells within our body. They are a double membrane, so we have two phospholipids. They are amphiphilic, so they have this hydrophilic or water-liking polar head and this hydrophobic or water-hating tails. So we have two fatty acids attached to this polar head, and we see this little kink in the fatty acid here. This is because it's got a double bond and it's a little more flexible. So we have these lipid bilayers that are around all of our membranes, but also around the lipoproteins that we just talked about. And these membranes really function when they're fluid and flexible. So depending on the type of fatty acids attached to the phospholipids, they can be more or less flexible. So lots more saturated fats, less flexible, more polyunsaturated, lots of double bonds and flexibility. We have much more flexible membranes. And this is important because we have a lot of proteins that attach themselves into the membrane, and they need to be able to move to function. And also we have proteins coming into the membrane, coming back out of the membrane. And we've seen that in the types of cases like obesity or high saturated fatty acid diets, the membrane starts to become more saturated and less flexible, and so we start to see problems with the utilization of like proteins within it. So, for instance, GLUT4 tends to work less efficiently if we've got very saturated, inflexible membranes. Also, one last thing with the phospholipids is that the fatty acids attached to them really are mediators of inflammation. I think Alan's going to talk more about that later on, but they're important that we have a variety of fatty acids attached to them so they can actually act in mitigating inflammation. And just to show you that they're not all working by themselves, they really are. It's a synergistic process. We have lipid membranes here with phospholipids. We've got polyunsaturates, which are working on inflammation. We've got fatty acids coming in through the membrane, being made into mono and di and triglycerides. They're going into the Krebs cycle here, the free fatty acids. They can also be attached to cholesterols and lipoproteins for transportation. So it really is a synergistic kind of balance for lipids within the body. So I'm going to pass back to Alan now, who's going to talk a little more about it.
Okay, thank you, Ellen. So there we go, guys. So Ellen was talking about monoglycerides, diglycerides, triglycerides, cholesterol esters, phospholipids. One of the important elements that we need to look at in all these various molecules is the fatty acid composition. Yeah, so remember what Ellen explained just now. Fatty acid is an acid, yes, it's a carboxylic acid, depending on the number of carbons, depending on the number of double bonds. That's when we see the characterization of each fatty acid. Humans can synthesize fatty acids, but not all types. There are two types of fatty acids that are truly essential: the omega-3 fatty acids and the omega-6 fatty acids. The omega-3 fatty acids, the first double bond is in the position 3 from the methyl group. Thank you very much. Sorry. And the omega-6 fatty acids, the first double bond is in the carbon 6 from the methyl group. Yeah? These are the truly essential fatty acids. So sources of omega-3 fatty acids include, for example, flaxseed, walnuts, chia seeds, rapeseed oil. These are important, abundant plant sources of omega-3 fatty acids. Whilst from the omega-6 fatty acid families, safflower oil, soybean oil, corn oil, these would be important dietary sources of omega-6 fatty acids. Yeah? These fatty acids, they are all elongated, they are desaturated, so the molecule becomes longer and it becomes more desaturated, which means enzymes remove hydrogens from the molecule, making it more flexible. Yeah, so removing hydrogens, so creating new double bonds in the molecules. So we reach this point over here in the pathways of elongation desaturation, where we see the eicosapentaenoic acid C25 and 3 and the arachidonic acid C24 and 6. Yeah. So the common denominator between these two fatty acids over here is the fact that they all have 20 carbons. So they're called eicosanoids. Yeah. So this term in Greek, eicosa, is for 20. Yeah. So both of them are 20 carbons. The difference lays here. So these fatty acid contains 3 double bonds, features 3 double bonds. Sorry, 3 double bonds. And the first double bond in the carbon 5. And this one over here features 4 double bonds, the first double bond in the carbon 6. Yeah. Dietary sources of omega-3 fatty acids include kelp. Kelp is a good source.
Yeah.
Kelp. You guys we don't have that in Brazil, but kelp and seaweed. These are like plants that grow in the sea. Yeah. And many people in Brazil call it algae, but this is a wrong term. It's not algae. These are like kind of plants that grow in the sea. So these are important sources of omega-3 fatty acids, but also fish overall. Freshwater fish is not as much as oily fish. Salmon, cod, sardines, these are important sources. On the other hand, from the omega-6 families, meat, dairy, chicken, eggs, they will be important sources of omega-6. These fatty acids continue to get elongated and desaturated, and eventually we see the docosahexaenoic acid over here, the docosapentaenoic acid over here. They have 22 carbons. These steps are far more complicated because they involve beta oxidation, and we're not going to cover that today. But the bottom line here is that this molecule, for example, the docosahexaenoic acid, is an important precursor for the synthesis of neuroprotectins, which are molecules associated with brain metabolism. So there we go. So very briefly, what we have over here, linoleic acid, if we count the number of carbons, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 carbons. The first position in the carbon 6, and there are 2 double bonds. Linoleic acid is elongated into arachidonic acid, 20 carbons now, with 4 double bonds. Docosahexaenoic acid, 22 carbons with 4 double bonds. Yeah. So the main sources of linoleic acid will be sunflower and rapeseed oil, whilst the main sources, dietary sources of arachidonic acid will be meat. So humans can get arachidonic acid either from the diet, or we can synthesize them ourselves. Okay. When it comes to the omega-3 family, what we have over here, alpha-linolenic acid, 18 carbons. The first double bond is in the position 3. Yeah. Flaxseed and walnut oils are important dietary sources of alpha-linolenic acid. alpha-linolenic acid can be then elongated and desaturated into eicosapentaenoic acid. So there we go. So 5 double bonds. Fish is an important source of the EPA, which then is eventually elongated and desaturated into docosahexaenoic acid. 22 carbons with 6 double bonds. The first double bond position 3. So humans can synthesize docosahexaenoic acid. However, the rate of synthesis is very low. And for that reason, arachidonic acid, eicosapentaenoic acid, docosahexaenoic acid, docosahexaenoic acid, they're considered as functionally essential fatty acids. The truly essential are these two, linoleic and alpha-linolenic. However, these are functionally essential because humans cannot synthesize them in sufficient amounts. That's why we have to have them in the diet. Yeah? So what we have over here, we have, Allain has shown in one of her previous slides a diagram that looks more or less like this, but far more complex. So what we have over here, we have various families of lipids. Yeah? And these families of lipids often overlap. Yeah? For example, the glycerolipids, why this term glycerolipids? Because there's a glycerol in the molecule. That's why we call it glycerolipids. There's a glycerol somewhere. Yeah? Within the glycerolipids, we have the triacylglycerol, which are used mainly for storage. Yeah, so they are also transported around the body through lipoproteins. We're going to talk a little more about that. And the typical molecules here. We have 1 glycerol and 3 fatty acids, as Allain explained. We have the family of glycerophospholipids. Yeah, so phosphatidylcholine, ethanolamine, serine, inositol, phosphatidylglycerol, cardiolipin. We're going to talk a little more about these phospholipids later. But the molecule overall is here. There is 1 glycerol, there are 2 fatty acids attached, and instead of the 3rd fatty acid, we have a phosphate head group, which makes this molecule more water-friendly. We have also within the families of phospholipids over here, the ether glycerolipids, in which the glycerol is replaced by a glycerol ether molecule. Yeah, so there's a bond over here. And this there is 1 fatty acid, and again we have the phosphate head group. The classical example is plasmalogen. So plasmalogens are very important in antioxidant properties for the cell. We have the sphingophospholipids. We have a sphingoside as opposed to a glycerol. Again, 1 fatty acid, a phosphate head group. So this molecule is more water-friendly, is slightly water-friendly, let's call it this way. And we also have the glycolipids. And within the family of glycolipids, cerebrosides, sulfatides, globosides, gangliosides, and they all have a variety of functions all over, in brain and in heart and in liver and all that. So we have a sphingoside, and instead of phosphate head group, we have a carbohydrate over here. Yeah? Right. Okay. So Allain showed this diagram to you guys a few minutes ago. I just want you to appreciate how busy the cell membrane is. Yeah. So just let's just appreciate this figure here for a moment. Yeah. So what we have over here, what I'm just hovering the mouse over the phosphate head groups on the outer layer, and then the phosphate head groups on the inner layer. Because of this specific arrangement, this side of the cell, the outside is water friendly, the inside is water friendly, but the middle is not water friendly, thanks to this specific arrangement. Yeah? And what we see over here, we see the formation of a vesicle. Yeah, so this process called budging. Yeah, this process of budging is very important when it comes to the cell secreting things, for example, or receive either in phagocytosis is the opposite, what you guys see over there. But this phenomenon is very important when it comes to membrane flexibility, because what Allain was talking about, the kink over here that we have in the fatty acids, if this kink is not there, the phospholipid becomes much more rigid. So it's much harder for this process of budging or phagocytosis, the opposite, to occur. So we have to have flexibility, and this flexibility is given to the membrane through the presence of all these many double bonds. We also have various channels, we have transporters, and we have hormones interacting with the cell membrane from the inside and from the outside. There are like some cell-cell interactions over here. So the cell membrane is very busy. So the phospholipid composition of cell membranes is paramount for cell signaling and cell survival. Yeah. So I'm going to talk very briefly about these 5 phospholipids here only: phosphatidyl, ethanolamine, serine, choline, sphingomyelin, phosphatidylinositol. Yeah. So they are all here: phosphatidylcholine, ethanolamine, serine, inositol. Only sphingomyelin is the kind of exception to the 5 ones I'm talking about. Remember that instead of the glycerol, we have the sphingoside, and then the fatty acids and the phosphate group. Yeah. Right. Okay. So first of all, we need to know that these phospholipids are not distributed across the cell membrane asymmetrically. The distribution is uneven. Yeah. Of course, we have 50% of phospholipids showing up on the outside, 50% showing on the inside. However, when we look at sphingomyelin, for example, sphingomyelin is far more abundant on the outside than on the inside. Around 20% over here for sphingomyelin on the outside, just about 5% or even less on the inside. Phosphatidylcholine is more abundant on the outside. Phosphatidylethanolamine abundantly on the inside. Serine is nearly 100% on the inside, and there is a reason for that, and I'll cover later. Phosphatidylinositol, we don't see a lot of it, but most of it is found on the inside of the cell. Yeah. So these 2 classes over here of phospholipids, sphingomyelin and phosphatidylcholine, they are naturally richer in saturated fatty acids than in polyunsaturated fatty acids. Whilst these 3 families over here, they are richer in PUFAs and poorer and unsaturated fatty acids. And the reason for that is that the insides, the inner leaflet of the cell membrane has to be more flexible than the outer leaflet. That's because of this process of budging, for example. So it's more important that the species containing more polyunsaturated fatty acids from the omega-3 fatty acids, sorry, the phospholipids containing more polyunsaturated fatty acids, they are present more abundantly on the inside of the cell. Yeah. So there we go. We can separate phospholipids, and once we separate phospholipids, we can analyze, quantify these phospholipids. A method I used to use many years ago is called thin layer chromatography. It's a very difficult, very laborious, and very expensive method. Yeah, so it takes a lot of time. But some of you may have already seen this type of picture over here. This is a thin layer chromatographic plate. So this layer is covered in like silica gel, and then we apply the phospholipids at the bottom of the plate and place it in a tank, and then the solvents migrate by capillarity all the way to the top, and the lipids travel along the membrane based on their affinity for the solvents. Yeah. So the phospholipid over here with lower affinity would be the lyso-PC, the lysophosphatidylcholine, then the phosphatidylinositol, phosphatidylserine, sphingomyelin, choline, ethanolamine, and then we have the neutral lipids over here. So what we do here, we can scrape the plate with a spatula and separate all these various phospholipids and then analyze them. Yeah. So what's happening here is that, let's go again, and we have the cell membrane, that's the outer leaflet, the inner leaflet. So I'm zooming in this part of the diagram over here. Yeah. So this is square over here. There's one phosphate group, the fatty acids, and this one over here. So I'm just showing it. It's slightly different over here. Yeah. So the glycerol is just over here. If you can count it, it's right over here. So 1, 2, 3 carbons. Look at that. 1, 2, 3 carbons, 2 fatty acids, and the phosphate group. These fatty acids, they are needed inside the cell. They are needed inside the cell for the synthesis of lipid mediators. For example, prostaglandins, thromboxanes, leukotrienes, marazines, lipoxins, and all sorts of lipid mediators. So the phospholipase A2, for example, is one of the phospholipases that hydrolyzes the free fatty acids over here from the phospholipid membrane, liberating the phosphate head group and the fatty acids. There is a huge family of phospholipases. Yeah, but the bottom line is that eventually the fatty acids become free. And this example over here, we have arachidonic acid, and arachidonic acid through the actions of COX-1, cyclooxygenase 1, or cyclooxygenase 2 can be converted into prostaglandin, for example, G2, and then H2, and then later on converted into other prostaglandins or other thromboxanes, etc. We have some drugs, for example, non-steroid anti-inflammatory drugs such as aspirin that will inhibit COX-1 and COX-2, or we have some COX-2 selective inhibitors, for example, COX-6. So there we go, folks. So this is a typical phospholipid. Over here we have the phosphated group. Let's have a look at the glycerol over here. 1, 2, 3 carbons. So there's the glycerol, 1 fatty acid over here, another one over here, and then there's a phosphated group. This part of the molecule gives a solubility to the molecule, makes it more water friendly. So phospholipases, they can act on the membrane, on the molecule at different sites. Yeah, for example, phospholipase 1, A1 will release this fatty acid over here from the first carbon in the glycerol. Phospholipase E2, phospholipase C will hydrolyze over here. Phospholipase G will hydrolyze in different areas. So there we go, folks. So, for example, in neural cells, phospholipase A2, which is this one over here, regulates an important balance between arachidonic acid conversion into pro-inflammatory mediator, and its reincorporation into the cell membrane. So lower or greater activity of phospholipase A2 will dictate the levels of arachidonic acid inside neural cells. This is a very important information. So when phospholipase A2 is increased, for example, we see more pro-inflammation. And then obviously this leads to increase oxidative stress, increase inflammation. So if we can reduce activity of phospholipase A2, we see then a reduction of all that. But remember, of course, that the composition of fatty acids is very important. So in conditions such as Alzheimer's, we see disturbances in this process. So what's happening here? Eventually, we want to get to this point over here. So contraction, depending on the cell, we see contraction, or we see like metabolic activities, we see transportation of molecules, hormones. We want to get to this point. We want to see responses in the cell. The cell will do something for us. So our cells are very specific. Each one of them will have specific jobs, specific tasks. Let's start from this block over here. So these are the cell functions. So how can we trigger the cell to do these jobs for us? Let's start from here, from messenger. So this messenger could be a hormone, for example. So I'm just going to give the example of vasopressin, antidiuretic hormone, ADH, antidiuretic hormone, also known as arginine vasopressin. It's a hormone that increases water absorption in the kidneys. So vasopressin will bind to its receptor on the cell membrane, and it triggers a cascade of intracellular events over here, characterized mainly by the phosphorylation of G proteins on the inner side of the cell. In the case of the alpha G protein, this one over here, when phosphorylated, activates phospholipase C. And phospholipase C, which is this one over here, phospholipase C, will then hydrolyze the phospholipids. For example, phosphatidylglycerol, releasing inositol 3-phosphate and diacylglycerol. Let's go back one slide. If I cut the molecule right over here, I have this part of the molecule free, and then this part of the molecule here free. One fatty acid, another fatty acid with a glycerol, I have then a diacylglycerol or diglyceride. Okay, we're going to talk about that in a moment. But there we go. This step is very, very important in cell signaling. Diacylglycerol, for example, we know that it can activate protein kinase C. When we see protein kinase C activation, and then obviously we see consumption of ATP, we see the formation of AGP, proteins get phosphorylated, and then we see responses in the cell. But at the same time, the inositol 3-phosphate, I'm going to talk a little more about it later, it can bind to specific receptors or transporters inside the cell, for example, in the endoplasmic reticulum, activating, for example, calcium channels. And these calcium channels will open, will increase the amount of calcium inside the cell. And every time we see calcium levels elevating, increasing inside the cell, we see a metabolic activity. Yeah, so calcium, when calcium is increasing inside the cell, it means that the cell is working somehow. Yeah, and then we see calmodulin activation, we see protein kinase activation, there we go, proteins are phosphorylated, and then we see then responses within the cell. Yeah, right. Okay, so let's just touch upon each and every one of these phospholipids very quickly, very quickly. Phosphatidylethanol. Okay, so this is the ethanolamine. So we have the glycerol over here, one fatty acid, another, and this phosphated group with 3 carbons. This is what we call ethanolamine. So if you remember, 1, 2, 3 met at 2 carbons met at ethanol, like ethanolamine. Okay, that's what it means. So phosphatidylethanol is very important across the cell membrane. Around 25% of, in a typical cell, around 25% of its phospholipids would be ethanolamine. However, in neural tissue, in spinal cord, for example, in white matter, this number increases significantly. Remember that in phosphatidylethanol, these fatty acids here, R1 and R2, they tend to be polyunsaturated fatty acids. So diet will play a major role in phosphatidylethanol composition, and for that reason, all the associated with all the functions that are attributed to ethanolamine. Oh, there we go. It's particularly rich in nervous tissue, the white matter, for example, nerves, neural tissue, spinal cord. 45% of these phospholipids will be PUFAs. Yeah, so they're rich in PUFAs, mainly in the inner cell membrane. They are present in the HA-rich receptor systems, for example, in the retina. In retina, for example, nearly 50% of the fatty acids in retina will be the HA. Yeah? And the reason for that is that the retina is so highly vascularized. Obviously, it's one of the organs associated with exposure to sunlight, and light can trigger oxidation. We know that well. Our skin is protected. We have various mechanisms that protect our skin, but the retina is essentially an extension of the system. And that's the reason why we have to have this extra layer protection, this extra protection. Yeah. So now with the phosphatidylserine, there we go, folks. We have the glycerol over here, one fatty acid, another fatty acid, and this part of the molecule is what we call phosphatidylserine. There's amino acid over here, serine. So phosphatidylserine is very rich in PUFAs, it's a substrate for nuclear receptors. And interestingly, the phosphatidylserine is found abundantly on the inside of the cell, on the inner layer, on the inner leaflet of the cell membrane. Okay? And then what happens is that during the process of apoptosis, phosphatidylserine is flipped to the outside, to the outer layer of the cell. Yeah, and there's an enzyme called lipase that is associated with this process. This is a very important signal for other cells, for surrounding cells, that when other cells, when neighboring cells identify phosphatidylserine on the surface, it means that that cell is going through the process of apoptosis. Yeah, so cell death, apoptosis. Yeah, there we go. Phosphatidylserine exposed on the surface. And interestingly, that many individuals, they buy, there is phosphatidylserine available in the market. Many people can buy phosphatidylserine, but my opinion particularly really is that I perhaps believe that people could be spending their money buying healthier foods, fresh fruit, fresh vegetables, and nice source of protein, so that they can have their fatty acids and also their amino acids from the diet rather than from the supplements. There we go. Right. So what about phosphatidylcholine over here? So there we go. We have, again, the glycerol, one fatty acid, another fatty acid, the phosphate group over here. Phosphatidylcholine is found abundantly on the outside, on the outer layer of the cell. It's rich in saturated fatty acids. It is very abundant on the outside. So phosphatidylcholine, you can also find it dissolved in blood. It's a phospholipid that it can be, because of this specific conformation over here on the choline part of the molecule. Yes, remember liver metabolism, choline. This molecule makes the phospholipid slightly more water friendly. And for that reason, phosphatidylcholine is a good transporter of fatty acids across in the body. So of course we have cholesterol, lipoproteins as transporters, but this phospholipid is a good transporter as well. Phosphatidylcholine is also an important trigger of cell-signaling events. Now phosphatidylinositol, we're going to talk a little more about them. So then there we go, folks. We have the glycerol over here, one fatty acid, another, and then we have the phosphate. The phosphate is a group, but interestingly, we have this phenolic ring over here. And this phenolic ring is so important for intracellular events. Okay, phosphatidylinositol is present abundantly on the inner cell membrane. It's a major second messenger. We're going to talk more about that. When this part of the molecule is phosphorylated, to phosphatidylinositol phosphate or bisphosphate and trisphosphate, it's involved, then it becomes involved in a range of intracellular events, such as lipid signaling, there we go, self-signaling, and brain trafficking, and all that. Now sphingomyelin, very briefly, over here. So there we go. This is the typical diagram. So we have this is sphingosine and then fatty acid and a phosphate head group. Through the actions of enzymes such as sphingomyelinase, we can convert into a ceramide, and then ceramidase, sphingosine, and then after that we can see sphingosine 1-phosphate. In this case, particularly over here, there's a phosphate over here, and this molecule is again very important for cell signaling. So it's a bilayer stabilizing lipids. Sphingomyelin is perhaps one of the most important phospholipids in the myelin sheath, that surrounds the axons in myelinated neurons. So remember white matter, the neurons are all myelinated. That extra layer of protection, myelin, one of the major phospholipids in myelin is sphingomyelin. It's a major component of the myelin sheath. There we go, folks. We have our axis over here, and all this surroundings over here will be layers and layers of myelin that will protect the cell, protect the axon, sorry. Is again the substrate for ceramide synthesis. Over here, there's a sugar, and which is a very important molecule associated with intracellular signaling. So there we go. So I have introduced all these phospholipids to you, but what do they mean and how can they change? So I'm going to start. I'll show a few examples of studies in which we can then quantify this phospholipids. We can analyze the composition of their fatty acids, and then we can see what is happening overall in terms of metabolic activity, diseases, etc., etc. This study over here, for example, is a study in which we recruited smokers and matched volunteers who were not smokers. So we have over here the control group, their age 27 years. The smokers, the age is 25, and this is statistically similar. Their BMI is also similar. Their diet is similar. This group over here, they smoke 20 cigarettes per week. If you think 20 cigarettes per week is not a lot, some people smoke 20 cigarettes per day.
It's like 3 a day.
Yeah, exactly. There we go. So it's 3. We're talking here about 3 or more cigarettes a day. When we look at the fatty acid composition of erythrocyte sphingomyelin, so I want you guys to have a look at this part of the table. Over here, the DHA, so let's just follow. So in control individuals, 4.6% of the fatty acids were DHA. In the smokers, we saw a drop to 2.56%. And this was statistically significant, which means that these fatty acids in these individuals are being mobilized to combat this excessive oxidative stress that's happening as a result of tobacco consumption. Likewise, we looked into, this is another study that we published a few years ago, looking into women with preeclampsia. You guys remember preeclampsia? So pregnant ladies with high blood pressure more towards the end of their pregnancies. So that's the typical plasmalogen over here. We have the glycerol ether, one fatty acid, and the phosphate group. Yeah. So this is plasmalogen. So when we hydrolyze the phosphate group over here, yeah, when we hydrolyze this part of the molecule, so we keep the glycerol ether and the fatty acid, we have something called dimethyl acetal, yeah, which is a product from plasmalogen breakdown. And then we can measure that, we can analyze that on the gas chromatograph. We can do that on the gas chromatograph. And then that's we have this C18-0 GMA, 18-0 GMA, 18-1 GMA, which means that this fatty acid over here is a C16-0 or C18-0 or C18-1. We can have all sorts of plasmalogens, okay? So it depends much on the composition of this fatty acid. Yeah? The more abundant ones would be C16, 18, 18-1. We have 18-2 GMA as well, so all sorts. But what we see over here, for example, in severe case of preeclampsia, we tend to see that the amount of plasmalogens tend to decrease in relation to the ladies who do not suffer with preeclampsia. Yeah. Okay, so over here, for example, the 18-0 GMA, which is abundant, in the control group, 7.87% of the plasmalogens were C18-0 GMA, and then we saw a drop to 7 to 21, which is essentially quite significant. Yeah. So there we go, folks. A little more about fatty acids. So some more studies over here that we've done a few years ago with ADHD adolescents, ADHD, attention deficit hyperactivity disorder. Yeah. So this work in this study, we recruited adolescent boys with ADHD, and then they provide a blood sample. We measured the amount of, we analyzed their fatty acid profile, but they were also subjected to psychometric tests. These psychometric tests are like questionnaires. Psychologists ask these very specific questions. They do all these exercises in order to assess the condition. One of the psychometric tests is to identify their capacity to detect emotions in others. Yeah. And then what we see over here, for example, of course, we see in this individual over here, this individual is showing a fearful face. So we can see this man is fearful. This is a fearful face. Yeah? So a happy face, a sad face, an angry face, a fearful face, or a face of disgust. Yeah. And then we measured all these indexes and we correlated them with their fatty acid levels. So we came up with this ICU score, the index of callous and unemotional trait. This callous behavior means a behavior in which the person does not have any empathy or sympathy. They cannot identify emotions. Yeah? This callous and unemotional traits. Yeah. So what happened over here is that as EPA, eicosapentaenoic acid, increased in their plasma phosphatidylcholine, this inability to detect people's emotions decreased. So they were more able to detect people's emotions when they had more EPA in their membrane, which means that with a higher amount of omega-3 fatty acids, these people, they showed behaviors that are more expected from humans, because as humans, we are expected to commiserate with other people's pain and suffering. Yeah? The same goes for DHA. Yeah? So the p-value over here is 0.54. Total omega-3, and interestingly, when it comes to omega-6, we tend to see a kind of positive correlation. Yeah. So the p-value over here is 0.81. 0.081. Sorry. Yeah. So of course, this is not statistically significant. However, it feels like it's going to become significant if we increase the sample size, possibly. Yeah. Which means that higher levels of omega-6 are more associated with this callous and unemotional traits in children. Now, Talking a little about Down syndrome now. Down syndrome children, they are at greater risk of developing Alzheimer's disease. Most Down syndrome children will develop Alzheimer's in their early 30s or late 20s. Unfortunately, it's a very difficult situation for the children, for the sufferers, and also for their families. And this is a study that we did a few years ago. We recruited Down syndrome children and their healthy siblings. The reason why we recruit their healthy siblings, like brothers and sisters, is because we wanted to make sure that the Down syndrome child had a diet that was as close as possible to the control group. Yeah. So we have over here, for example, in the Down syndrome group, we had 9 children in total. It was a small study, but that was a very difficult study. This study was very difficult because it was difficult to find the Down syndrome children, but also their healthy siblings. Yeah. Remember that children living under the same household, in the same household, it's likely that they have the same diet or a very similar diet. And then, of course, we analyze their food diaries, and indeed, their diets are similar. And then what happened over here, we did mass spectrometric analysis of these phospholipids. And what we observed, we isolated the phosphatidylcholine molecular species. And that's when we look at the we look at the phospholipids in its entire composition, right? And then we have over here an increase in species with abundance of saturated and monounsaturated, and a decrease in the abundance of species containing polyunsaturated fatty acids. So in summary, what happens over here in Down syndrome, we see an elevation of this molecule and a decrease in this molecule. So over here, so look at that. All these unsaturations are important for membrane flexibility, for the synthesis of lipid mediators we discussed earlier. Yeah. So previous studies again in London, we found that phosphatidylcholine containing DHA, for example, is decreased in Alzheimer's. We saw our p-value was 0.08. So it's well possible that in the future, these children will show the same phospholipid profile as Alzheimer's patients. And there we go. So there we go, folks. So phospholipids and fatty acids have been, in a way or another, associated with a range of psychiatric diseases. For example, bipolar disorder, depression, major depression, schizophrenia. So we have also papers that looked into mild cognitive impairment, Alzheimer's disease, and their correlation with fatty acids and phospholipids. Schizophrenia, again. So borderline personality disorder, post-traumatic stress disorder. So they are in all these psychiatric diseases or disorders, we have, in a way or another, some sort of involvement, with fatty acids. And more specifically, phosphatidylinositol related pathologies. This is a new field of research, and we believe that phosphatidylinositol is heavily involved in a way or another in the development of psychiatric disorders. Yeah? So we have over here a phosphatidylinositol, and it is phosphorylated. We have 1, I cannot see, 4 or 5. We have this phosphates over here that makes the molecule phosphorylated. These enzymes over here, the phosphoinositide 4 kinase, is the one that inserts the phosphate in the phospholipid. So what we have over here, we have this phospholipid. Look at that, 1 fatty acid and 1 fatty acid phosphatic group. The phenolic ring, carbons 1, 2, 3, 4, 5, 6. This is our PI phosphatidylinositol. Yeah. This phosphate over here is what holds the diacylglycerol to the phenolic ring. This is a phosphodiester bond. And this part of the molecule is inserted into the membrane. Normally, these fatty acids are arachidonic, normally stearic acids, normally raw, but depending on much of the diet. Yeah? And then what's happening here is that This phosphatidylinositol is phosphorylated. Look at that. We see the introduction of phosphates in positions 4 and 5, and these enzymes are the phosphodiinositide 4 kinase and 5 kinase. They are responsible for the insertion of these phosphates. Yeah? And this one particularly over here, the phosphodiinositide 3 kinase is involved in a range of other, is associated with cell proliferation, migration, and etc. So let's see in this diagram, let's see what happens. We're going to see eventually the production of diacylglycerol and inositol triphosphate. So remember, we have the phosphatidylinositol across the membrane. This phosphodiester bond is hydrolyzed, releasing the inositol triphosphate, which goes up here, and the diacylglycerol that goes down here. Yeah. Oops. And there we go. So, and then, for example, a hormone, like as the example I gave before, vasopressin is phosphorylates protein G, which activates phospholipase C, releasing the inositol triphosphate. And then what happens after is that the inositol triphosphate, it can either increase or decrease the number of phosphates depending on enzymatic activity inside the cell. Yeah? So we have, for example, phosphatases. These phosphatases are enzymes that remove phosphate groups. So we have over here our inositol triphosphate. Through the action of phosphatases, we see bisphosphate or phosphate. Yeah? So phosphate, which means the inositol without any phosphate whatsoever. But if we have kinases, these kinases will add phosphate groups, forming from triphosphate, tetra, kisphosphate, or pentaphosphate, etc. So inserting another an extra phosphate into the molecule. And this is very important because the amount of inositol triphosphate inside the cell is an important is very important to induce or not induce cell activity. So if there's more triphosphate, it means that the cell is working harder. If there's less, it means that the cells will work a bit less. Okay, so this is a very important. The levels of inositol triphosphate inside the cell are very important. Yeah. So our inositol triphosphate is right over here, and through a range of enzymatic reactions, we can see more or less phosphorylated, and then depending on its levels inside the cell, and then we see more or less cell activity. Yeah. Diacylglycerol is also very important when it comes to cell activity. If we go back over here a moment, this is what we're talking about now, diacylglycerol. And let me talk a little about diacylglycerol in a moment. And there we go. Diacylglycerol is very important because it activates protein kinase C inside the cell. And with that, we see a greater affinity for calcium. There's an increase in calcium inside the cell. And there we go. What we have over here, the phospholipid C, hydrolyzing the phosphatidylinositol, releasing inositol inside the cell that will do various jobs, but the diacylglycerol is here. Yeah, this diacylglycerol will be so important for example, for increasing enzymatic activity inside the cell. Diacylglycerol, we have a very short life, it's just a few seconds. It will very soon be phosphorylated to phosphatidic acid, and then we see then if it's phosphorylated to phosphatidic acid, we see the formation of there we go, phosphatidides, and then if not, we see glycerol and fatty acids. Yeah, so which will be important for pathway. Right. Okay. So this is what Ellen was talking earlier about the importance of analyzing these fatty acids individually, these fatty acid families, these lipids individually, because when we analyze the full composition of total lipid extract, we don't see differences. But when we split them up and analyze them individually, we see differences. And this is a paper that my colleague Dr. Boldarin did with. Boldarin came to Worcester, spent some time with us here from Brazil, and then we worked quite a lot and produced this paper. Over here, that I'm going to talk in a moment. But the bottom line of the paper is that the adipocyte is an altruistic cell
in the sense that it stores fatty acids, but when there is a need, the cell will release the important fatty acids for use elsewhere in the body. Yeah, for example, fasting, starvation, etc. Yeah. So Allain showed this slide earlier. This is a cholesterol ester. That's a major component over here, the cell membrane. And then what we have, if you guys compare these 4 diagrams, the fatty acid composition of cholesterol esters can change. Yeah. And then when there's dietary provision, triglycerides are stored inside the cell. When there's a need, these triglycerides will be hydrolyzed into free fatty acid and glycerol. And then this free fatty acid can be bioconverted into a cholesterol ester, which travels across the membrane and can be released. Yeah? And then it will travel, over around blood circulation, lipoproteins, for example, providing structural needs for prime tissues such as brain, heart, etc. Yeah? So there we go. So what we have over here, this is our collaborators. I'm going to pass it on to Allain in a moment. Dr. Eliane Ribeiro over here, Dr. Lila Oyama, Dr. Claudia Oler, Monica, my colleague Monica over here. This is me like years ago. And there's Allain and Walter here. And then this is my colleague, our colleague, Dr. Hirata. And it's your turn?
Yeah.
Okay, so I'm going to pass it on to Allain, and then Allain will deliver a few more slides now.
So just to go back to our collaborations, both with Dr. Tellis, our wonderful host, and Dr. Ribeiro, they look at high-fat diets. And what we see with these high-fat diets is that we have decreased membrane fluidity, impaired insulin signaling, as well as disrupted glucose homeostasis and increased oxidative stress and inflammation. So going to kind of the obesity prevalence in Brazil, there is nearly 55% of adults are either classified as overweight or obese. So there's a real need for kind of research into this area, with almost 20% of adults identified as obese. And that number actually increases when we come to the UK. We actually have 63% of the population which is classified either in the overweight or obese category. And this costs our health system billions of pounds, and it's increasing all the time. And it's often associated with this high-fat Western style diet. And like I said in the previous slide, it has a host of issues that come with this high-fat diet, from the altered GLUT transporters to the decreased fluid membrane, dyslipidemia, and increased amounts of oxidative stress and inflammation. So again, saying before, even if we look at the total fatty acids from an entire sample, we don't always get to see the specifics of what are changing. So we really had a need in our group here to take the samples and separate them out into triglycerides, mono and diglycerides, cholesterol esters, and phospholipids. So we could really see the dynamic changes that are happening within the cell. So we took resources from the Lipid Maps website and organization, and we identified in the literature that we could use solid phase extraction. So this is a silica gel column chromatography. So we could take our sample, load it onto a column, and using different polarities of solvents, we could then elute different amounts of the sample. So we were looking at mono and diglycerides, triglycerides, the cholesterol esters, and the phospholipids. And so we used silica-based columns, and we used standards commercially available so that we could test and optimize this process. And we used this paper Ingalls as all as our original starting point. We verified it through both GC-FID, but also by HPLC analysis. So we took egg yolk, which is rich in phospholipids, but also cholesterol and other fatty acids, separated on the column, and then we put it through our HPLC method for phospholipids, and it gave us a really clean sample of phospholipids. So we knew that our method was working. So this brought us back to our collaborations with Walter, who came to visit us. And so he produced 2 different papers from the samples that he brought with us here. And what we saw from the original paper here was that the total fatty acid composition didn't really change. And so we didn't see huge changes in the total fatty acids. So we then put his samples through this separatory silica columns, and that's really where we started to see the changes. So I'm aware we're a bit, we're getting close to time, so I'm going to whiz through these. So if you do want to have a look at the study itself, you can get these 2 papers and have a look through. But essentially, Volter's research is looking at high-fat diets and HRT treatment in rats. And so when we, so there was a whole pile of dynamic changes that happened in a lot of the metabolistic products in the rats. But what we were really interested in was the changes in the fatty acids. So once we separated out the samples, we started to see changes in the cholesterol esters, particularly in the saturated fatty acids. And you can see here that between the control groups and the groups that had estrogen treatment, we really started to see dynamic changes in the saturated fatty acid, like a decrease of like 36%, just by that we had not been able to see when looking at the total fatty acid composition. And again, we start to see changes in the monounsaturates within the cholesterol ester fraction itself. Again, we saw changes, a lot of changes within the polyunsaturated fatty acids that were attached to the cholesterol esters. And again, you can see here, just by my highlighting, we really started to see these changes, which again were completely masked, but when we looked at the total fatty acid composition. So again, just to highlight here, you can really start to see these changes. So a huge increase in C20, 4, and 6 from the control group as compared to the operated rats that had not had estrogen. And again, we saw really a lot of changes within the polyunsaturated fatty acids. Again, and really, I'm just trying to highlight here that this is from having total saturated fatty acids. We did not see this, but by separating it out and looking at the different lipid classes, this is really where we start to see the dynamic changes within the cell itself. Do you want to?
Yeah, there we go. So there we go, folks. Just to re-emphasize that the need for fatty acids in prime tissues is dictating what happens inside the adipocyte. So if these rats, for example, what's happening over here, we see this is the control rat, a rat that has been ovarectomized, and a rat that has been ovarectomized but received estrogen. So you see over here that ovarectomy increases the omega-3 in the cholesterol esters, but then ovarectomy plus hormone replacement therapy decreases that, which means that when we look at this molecule over here, this molecule is richer in omega-3 in the rat that was ovarectomized, but not because this is good, because it's bad. Simply because the fatty acids, the fatty acids stored inside the cell, inside the adipocytes, they get immobilized, they'll be exported, and they will travel to prime tissues. But with the hormone replacement therapy, we see a decrease of that, which means that there is a lower need for omega-3 to prime tissues coming from cholesterol esters. Yeah. So there we go, folks. So, and then we see this profile gets repeated again and again and again in obese rats. Their metabolic profile is very different as compared to a rat that had a controlled diet. We do not see the very many changes, but to begin with, the level of omega-3 is significantly smaller, significantly lower, I beg your pardon. Yeah, so there is far less essential fatty acids in the rats fed a large rich diet. Yeah. So there we go, folks. So in summary, a large rich diets, they deteriorate fatty acid profile.
cholesterol ester, there's an error here, sorry, is an important component of some brain, exports fatty acids via lipoproteins to tissues, and then there we go. So this study has many limitations, but the take-home message here is that in menopause we see aging, of course, changes in the central system, peripheral and endocrine systems. Estrogen replacement therapy is helpful, but alone does not fully restore metabolic profile. Yep. So, and then Ellen will, is your turn now?
Yeah,
So
I'm going to whiz through this.
Yes, so we are aware time is now 16:20. We have 10 minutes to finish. So Ellen will just touch and then we'll be able to answer questions later.
Yeah, so for those who joined us at the very beginning, you may recognize this lovely person. This is our host. And so we've had a collaboration between Dr. Monica Tellis and her PhD student, now graduated, Dr. Bruna Harata. So she came to us for a year. And so they've worked a lot on ginkgo biloba in high-fat diets, and they've published a lot of papers. So I do recommend you go and read them if you haven't seen them before. But just to briefly go through it, they use Worcester rats that are fed a normal fat diet or a high-fat diet. And then they treat the rats with either ginkgo or saline. And they've collected tissue samples from the rats, and then Bruna, when she came to visit us for a year, she brought her samples with her, and we did a lot of work with them while she was here, and then we've worked on them since. And so just to, you can't really see here, but we have a lot of adipose tissues, as well as liver, plasma, and brain tissues. We analyzed the composition of the chow that the rats had here. So again, GCFID, we analyzed and really to start to see the changes. So there's, if you can see here, there's a lot more saturated fatty acids in this high fat diet, a lot more monounsaturated fatty acids compared to a normal fat diet, which tends to have more polyunsaturates and less saturated fat. So these rats already are exposed to higher saturated fats and mono compared to the polyunsaturates, which will mitigate inflammation. So again, within the study itself, they've seen body weight goes up with a high fat diet, but with ginkgo biloba, the energy intake, it starts to decrease when they're treated with the ginkgo. So when we took the lipid samples from all the tissues that came from this study when Bruna came over, we first looked at the total fatty acid profiles. And just to give you an example, this is the adipose tissues. We've got retroperitoneal, mesenteric, and epididymal tissues. And we didn't really see a huge amount of changes. So these are the fatty acids. We don't really see changes here of the saturate fats. We see some changes in the monounsaturates, particularly in the mesenteric. But again, we didn't see huge changes in, say, the polyunsaturated fatty acids. So we then used our method of separating them out into the different classes of lipids. So they went through the silica columns, they were extracted using the different polarities of solvents. And this, again, so we looked at mono and diglycerides, the cholesterol esters, the triglycerides, and the phospholipids. And this is really where we start to see the changes. So when we're looking at the triglycerides versus the mono and diglycerides and the cholesterol esters, we really, this is mesenteric adipose tissue. So remember, we saw some changes in the total fatty acids, but not huge amounts. But now that we're starting to look at the different classes, we're really starting to see the dynamic changes. So again, we're not seeing a lot of changes in the triglycerides, but we are starting to see in the mono and diglycerides and also the cholesterol esters, particularly in the saturated fatty acids and the total amount of saturated fatty acids. We really start seeing changes that we did not see just looking at the total fatty acid profile. When we looked at the monounsaturated fatty acids, again, we start to see changes in the mono and diglyceride fractions, but again, now we start to see changes in the triglycerides for both kind of the total amount of monounsaturated fatty acids and also the amount of omega-9 fatty acids. When we looked at the polyunsaturated fatty acids, again, this is when we started to see the dynamic changes in the mono and diglyceride fraction and the cholesterol ester fractions, particularly for the N6 polyunsaturated C18, 2, and 6. And this also, we saw changes with the omega-3 polyunsaturates, which again was completely masked by just looking at the total fatty acid sample. So this was really great news, because if I quickly go back to what you can see here with Monica, and they really did see lots of changes in, say, the epididymal white adipose tissue, in the proteome of the retroperitoneal, but we weren't seeing it when we were looking at the total fatty acids. But by breaking it down, this is where we really started to see where the changes were occurring. So this kind of, it told us we were on the right step, and by separating them out, it really was helping us understand the changes that were happening in the rats, which we weren't able to see just looking at the total samples. There we go. And again, lastly, when we looked at the phospholipids, this is where we really started to see the changes. You can start to see stars coming up all over the place. We saw huge amounts of statistical changes in the phospholipid fraction of the mesenteric tissue. So we could really start seeing that the signaling that we would expect to see in the phospholipids, that the lipids are changing, the fatty acids associated with those phospholipids that Alan spoke to you about earlier. We really start to see where the changes are occurring within this fraction, which really excited us, because it kind of gave us a lot of new avenues to go down in terms of the research that we're looking at. So just to conclude, looking at total fatty acids is still a great benchmark to start with. But if you don't see the changes, but you expect that you might see, if you're looking at different proteomics of your samples, it might be worth separating them out and looking at the different classes of the lipids to really see what's happening within the classes itself. And finally, from my side, I really want to thank my supervisor, Alan here, and my co-supervisors, Dr. Cherry and Dr. Coles, and all of our technical support team here, as well as all of our collaborators at the University of Sao Paulo, Dr. Rubiro, Dr. Tellez, Dr. Hirata, Dr. Bouldering, and their technical team, because my PhD wouldn't have happened if their research hadn't have happened and we hadn't have done that collaboration. And again, thank you to all the supporting funding agencies that have contributed to this research.
There we go.
Thank you guys. I think that was all. It was just on time, 16:28, so we have 2 minutes for questions.
That's exactly. So thank you very much for the wonderful presentation, and also for the wonderful results that they were new for me. So we will talk later about them. I'm very glad to see all these wonderful results. And I will address you one question because we are run out of time. So Maria Isabel, our colleague, is asking.
Hello.
Yeah, she says, thanks, Alan and Ellen, for the great presentation. It was really, really nice. So she's asking, recent reports have uncovered the role of individual lipid species on membrane alter toll-like receptor signaling, endocytosis, and cytokine secretion. How difficult is to identify the function of individual lipid species or the composition of membrane lipids in processes like that? Because particularly, I'm very interested in this tissue.
You're talking about toll-like receptor.
Yeah.
Well, so there we go. The composition for the phospholipids is very much important for the way the cell responds to attacks. For example, the toll-like receptor 4 will be activated when there is a pro-inflammatory environment. Yeah. So, and the toll-like receptor 4 is anchored across the membrane of the cell. So if the membrane is more or less fluid, this will certainly impact all this molecular, there's some molecular configuration of the way that the receptor is anchored. And if the membrane is more or less fluid, we will certainly should be able to see a more or less dynamic response from the cell. I don't know if I have answered the question. Have I?
I think so. Maybe Maria Isabel will write something here. I think we have a time for another question.
Can we read?
Yeah.
From Joel, it's our colleague from Unifesp. Do you want me to read, or maybe quickly, if you read?
Joel, here over here, hold on a second. Lipid droplet accumulation seems to be directly involved in the establishment of different types of diseases. Yes, for example, liver, of course, different types of cancer development. Could you talk about the role of lipid droplets in the regulation of cell signaling? These lipid droplets, of course, these lipid droplets involves, I think I need to understand a little more what you mean by lipid droplets, because you can have intracellular accumulation of lipids. Yeah. So like my cells, for example, I don't know whether you asked about my cells, or are you talking more about accumulation of fatty acids or like fibroblasts or adipocytes in tissues that shouldn't have that many, for example, liver tissue? Is what are we talking? Are these lipid droplets inside or outside the cell?
So, thank you again for your talk. I'm very glad. You're waiting for Joel, right?
What? Sorry.
I think you answered the question already, right?
Yeah. I'll be available via email. Monica, welcome to send our email addresses. I think we have our email address.
Yeah, they're on the website. So if they look our names up, they'll find us on the university website.
Okay, and I can make the contact between our colleagues and you. So thank you once more for your wonderful presentation. It was, especially for me, it was a great pleasure, and I'm sure that for all of the participants, I'm sure they enjoyed a lot. I can see by the chat, for the comments in the chat, and also in our group, we have a special group for the organizing committee. So they are all saying wonderful things about your talk. So thank you very much. In the name of the organizing committee, I want to thank you again, and I hope to see you soon. I'm missing you guys.
And thank you for having us.
And Ellen, I'm waiting for you in Brazil.
I know. It's on my to-do list.
Hopefully soon.
Ellen will be our host. We're going to introduce her to the typical Brazilian food, and we'll write many papers, Monica.
Well, you will have to spend at least 2 years here with us.
I'm gonna send Ellen over for a postdoc with Monica.
Perfect. You will be very welcome here, Ellen.
Thank you.
Okay, so thank you very much. Now we have a lunch time, and we meet again at 2 o'clock, right? So thank you, and guys, thank you again.
Thank you.
Thank you.
All the best for you.
Thank you very much, guys. Bye-bye. Thank you.
Bye.
Okay.
Thanks,
Bye-bye.
Monica.
Thank you.